Hi, this is a short video on DNA profiling and just the basics on how it works. And I'm using gizmos to demonstrate this. DNA profiling is based on just a section of DNA from organisms. DNA profiling is used in a lot of crime or forensics cases to solve crimes. It's used in for paternity tests. It's used for also identifying relatives in the case of inheritances. And it was used in the identification of some of the victims of 9/11. And we're going to go to this little forensics lab. And here we're looking at a section of DNA. It's showing gene A, gene B, gene C, and then in between, you may notice it has a non coding region. So 99% of our DNA is alike in one species. Really, DNA fingerprinting depends on the use of these non coding regions of DNA. So, for all of your regions like gene A, gene B, gene C, they have all of those nitrogen bases that code for proteins, and your ribosomes are producing proteins as a result of those instructions. Then you have sections that are in between that don't code for anything. Um, if we click on this non coding region, it's going to show us the DNA of three different people. And if we look at the strands of DNA for these three different people, one thing that is unique to each individual is the length of the non coding region. So you may notice person one has a much longer non coding region here than person two and person three. Another thing that is going to be different about these non coding regions in these three different people is you're going to have repeating patterns. Okay. So each person has the same pattern, but person one has the repeating pattern four times in that section. These are called short tandem repeats. So person one has four short tandem repeats. Person two has two short tandem repeats. Person three has three short tandem repeats. So those are the major differences. And so if we're going to compare DNA of three different people and we want to look at that small percentage of DNA that's completely different, it's going to be in the non-coding regions. Okay. In order to view these non-coding regions, we have to copy the DNA, and we do that using what's called primers. Okay. And so those primers are just on the ends of the non-coding sections, and we have DNA polymerase coming in to make copies of that DNA strand. Those are enzymes that are making copies. So the DNA is copied a million times. And again, we can look at each person and see that they have the different lengths in the non coding region. Now, because they have different lengths, you can see again, it's showing the three people with the three different lengths. We're going to insert those copies of the non coding regions into this electrophoresis, this gel tray. And we're going to use an electric current gel electrophoresis to separate the strands. So once that electric current is put into or the gel pan, okay, each different section is going to travel a different distance from where it started. And so you have person one showing this band, person two and person three. They've all traveled a different distance because person one had the longest DNA, or that longest DNA strand. So it didn't travel as far as the shortest DNA. And that's really going to show you those differences on the gel pan. Okay, if we go back to our just our regular our gene sections, start again here. If we go back and we look at the sections of genes, okay, there really doesn't appear to be any difference just if you're looking at these particular strands. You may note here that most of these nitrogen bases are going to be exactly the same. There is one slight difference in person two. You can see right here, you have a different pair of nitrogen bases. And that's a slight difference. That's a slight mutation. If you had major differences in the gene, there would be a genetic disorder or there would be a health issue. But looking at the coding regions of the DNA, you don't have any short tandem repeats. You don't have the different lengths. And so if we were to go through the process of using the primer and copying the DNA on all three of these strands, you're not going to have that, you're not going to have a notable difference. If we look at this from afar here, they all have the same length. There's no short tandem repeats. And if we put that in the gel pan and using electrophoresis, it's just going to show us that the DNA looks the same. So the premise behind DNA fingerprinting is the use of the non-coding regions. And if we're using the non-coding regions, we're able to compare DNA profiles.